notes. Okay. Is this thing on?
Mm-hmm.
Yeah, it's running. Okay. There's markers here too.
Okay.
Okay.
Yo. we're just gonna get this thing done. So I'm just gonna do the first question, and I'll just read it off. Yeah. Do I have to say who's in this? No.
Yeah, we can introduce each other when we come up.
Okay.
Yeah.
So, so for part A, it says this question concerns a human disease called Angelman syndrome, which displays genomic imprinting. In the case of Angelman syndrome, the imprint silences the paternal inherited alleles. Suppose little a, is the Angelman syndrome allele, while big A is the non-disease allele. The following cross is done with a male who is heterozygous for the allele, with a female that is also heterozygous for the allele. What are the genotypes and phenotypes of the progeny? So, if we just drew out the little square, we have the dad, we have the mom. So if we just, yeah, that's right. We just do it like normal. That's our Punnett square. However, the paternal allele is silenced, so it doesn't matter. So the way I understand it, everything is based off of the mother. So whatever the mother is, then that is going to be, that is going to tell if the future children have the disease or not. In this case, none are diseased, if I spell that right. However, the genotypes are still like normal. Homozygous dominant is 1 out of 4. Heterozygous is half. Homozygous recessive is low. Yep.
So the mom gives big A to 2 of them. So 2 of them are going to have the disease.
Wait, what? Oh.
It's okay. Yeah. That's all. That's we're all working together. Yeah.
We're on track.
It's okay. Yeah.
So then 2 of them have it. Just kidding. We probably should have talked about this before we did this. So 50% then. Anyway, for B, this question concerns the situation presented in part A. The following cross is done. So let's just do another one. So, here we go. So we have the dad again, heterozygous mom. Mom actually has it. So, wait, this is the answer for part B.
Yeah.
I was doing A. I was doing part A in the first one.
Yeah.
No, that's the answer for part A. For part B, it'll be 100%.
Oh yeah, that's right. That makes sense. Yeah, just kidding. Since mom has it, everybody's gonna have it.
Yes.
Yeah. Okay. Sorry, guys.
It's okay. Yeah.
So yeah, we have the genotypes are 50%. Yeah, and then everybody's gonna have the disease since mom has it. That's supposed to say disease. So going on to part C, suppose a woman has triple X syndrome. However, the X6, that's what I call it, has been deleted from 2 of the X chromosomes in each of her cells. How many bar bodies are found in each of the woman's cells? So a woman has 3. We can get rid of all of this. So if a woman has this type of syndrome, she's going to have 3 X chromosomes. However, what does it say? If 2 of them deleted, then in a way they're pretty much like non-existent. So we only have one X chromosome left. And because there's only one X chromosome left, that one's only functioning, meaning that there are 0 bar bodies. Yeah? That's the way that I look at it. And then for D, suppose you isolate a cell from wait. Oh yeah, that's the same one. Suppose you isolate a cell from a female cat with 2 X chromosomes. However, the T6 genes on each X chromosome are non-functional, while the exist genes on each X chromosome are functional. How many bar bodies are expected to be found in this cell? So the way that I understand it is that let's see. The way I understand it, T6 is the one that I guess turns on the X chromosome. I think that's the way that I said it earlier. Turns it on while the exist gene is the one that turns it off. Something like that. Basically, let's see. Either way, in the end, there's going to be two bar bodies that the mom is going to have. Can you actually explain that a little bit? I lost my train of thought.
Sure, yeah.
Yeah.
So so the T6 genes on each chromosome are non-functional. So what the cysts do work, and the cysts are the ones that deactivate the X chromosome, but the T6 are the ones that keep it active. Since the T6 are inactivated on both chromosomes, 2 bar bodies will form.
Yeah. And that's it for number 1.
Okay.
Just like cover this.
All right, good job, Eric. All right.
200 number 2.
Yeah. Okay, so for number 2, the first it's just fill in the blanks. So for A, blank is an example of a haloandric gene. So haloandric means it's only one of them. So if we remember,
Oh yeah, sorry.
It's okay. So if we remember, you have a chromosome. That's my really bad drawing of a chromosome. And it has a homologous pair. And then on each chromosome, there will be a gene. So there's this gene, and then it's homologous pair, two genes that both code for the same trait, but they could be different alleles of the same gene. Well, if it's haloandric, that means this gene does not have a homologous pair. There's only one gene. So SRY is a gene that's found on the Y chromosome, but is not found on the X chromosome. So males only have the one. So that's an example of that. And then also, blank is an example of a gene found on both the X and Y chromosomes, MIC2. A cross is made between IGF2 and IGF2, minus male mouse, and IGF2 and IGF2 minus female. What are the phenotypes of the male of the offspring? So my answer was half will be normal body size, and the other half will be dwarf body size. And why did I say it was because IGF2 is deactivated in one of the I don't remember why it was like that. It's because it's deactivated in the maternal. Yes, deactivated in the maternal parent, which means only the paternal is expressed. So since he is half the regular and half is dwarf body size, those are going to express themselves in equal proportion of 1:1, which is why I gave the answer. Okay, so part C. in the blank pattern of inheritance, an allele can be dominant in one sex, while the same allele is recessive in the other sex. And that was sex influence. Like, for example, in humans, the trait for balding is dominant in males, which is why so many men are bald, and it's recessive in females. Okay, so part D. Blank is an epigenetic process that occurs during gamete formation. That's genomic imprinting. That's where that's where genes are silenced. While blank is an epigenetic process that occurs during embryogenesis. That's dosage compensation. That's like where the females have two X chromosomes and one of them is silenced. Okay, one of the topics discussed in lesson, well, mine says 2, I think it's supposed to be 3. Yeah, involved the formation of a double-stranded RNA molecule. What two molecules form the two halves of this double-stranded RNA? They are cis RNA and T6 RNA. Yeah, so then the last part, I think it's the last part, is the last part. When reciprocal crosses give identical results, the alleles that control the trait in question are located on autosomal chromosomes. Because they're on autosomal chromosomes in the pre-cross, they will be identical results to the parents. When reciprocal crosses give different results, the alleles that control the trait in question are located on X chromosomes. So, so yeah, that's how that works. Ready?
Yeah. Okay. Cool. So we're going to go to question 3. So for question 3, we have a woman's father has OTD, an X-linked recessive disorder producing mental deterioration. Sorry, I'm like, if not properly treated, the woman's mother is homozygous and is unaffected. So for part A, it wants to know what her mother and her father would be. And so for this, it would be really bad. Her father is going to be X minus and Y, and then her mother is going to be XX with no minuses. And so for part B, we have if the woman has a child with a man who does not have OTD, what is the chance that their first child will be a son with OTD? And so what I did is I made a cross with them. So X minus Y and then XX. There we go. And so with this cross, half of the females will be unaffected, and then half of the males will be unaffected. But
this is assuming that the mother is
homozygous.
Is homozygous.
And so if we don't assume she is homozygous, we assume she's heterozygous, we're going to get a different cross. So you can see that the body's in the way. So you have X minus. And so from that, we're going to have different phenotypes because we're assuming that she's heterozygous instead of homozygous. And we'll have is it 1?
It's 1 out of 8. Yeah.
1 out of 8. So in the end, we'll have a 1 out of 8 chance that they'll have a son that's affected. And so for part C, we have identify the genotype of a male with whom the woman could produce a daughter with OTD. And so for this, I said the father would have to be affected and the mother would have to be a carrier or affected. So we don't know if she is homozygous or heterozygous to have an affected daughter. And so from that, we're going to do basically the same cross. If we just put in little minuses right here. So this is her being affected. Her being affected and her husband being affected. And then here we have her
As a carrier.
as a carrier. Thank you.
It's okay.
And so from that, we can see that they are either going to be half of, or 2/4 affected females and then a half affected males, or for option B, we have 1/4 unaffected females, 1/4 affected females, 1/4 unaffected males, and 1/4 affected males. And so that'll take us to question 4.
Okay. All right. So for question 4, the presence of a beard in some species of goats is determined by an autosomal gene that is dominant in males and recessive in females. So just to explain, if it's dominant in males, that means he can be if it's dominant in males, he can be homozygous dominant or heterozygous and have a beard. But since it's recessive in females, so this is for male, since it's recessive in females, the female would have to be should have to be dominant, homozygous dominant to have the beard. If she were heterozygous, she would be unbearded. Yes, because the unbearded trait is dominant in the female. Okay. So part A, suppose that a cross produces a 3 beard to 1 no beard phenotype ratio among male offspring. What are the genotypes and phenotypes of the parents? Okay, so let's walk through this. So, I have 2 hands. So the dad is going to be big B, little b, and the mom is going to be also big B, little b. And if we do a cross, we get okay, so if we do a cross, we get 1 that's unbearded and 3 that are bearded for males. And so the father is going to be bearded and the mother is going to be unbearded. And then part D, suppose that a cross produces a 1 beard to 3 no beard phenotype ratio among female offspring. What are the genotypes and phenotypes of the parents? So actually, the same cross works here because this cross gives us a 3 to 1 ratio for males, but this would give us a 1 to 3 ratio for females because she'd have to be like this to be unbearded. But these 3 are going to be
Should be unbearded.
I'm sorry, yes, they're sorry. So these 3 are going to be unbearded, and only this one will be bearded among females. So same as above, same as part A. And part C, suppose a cross between a male and a male with a beard and a female without a beard produces a 1 beard to 1 no beard phenotype ratio among the female offspring. What are the genotypes of the parents? Okay, so dad is going to be big B, little b, and mom is going to be, oh, actually, what is this? It's going to be big B, big B, which will give us, that will give us, sorry, I'm running out of room here. Okay, and that gives us, this is among male, among female, yeah. So 2 will be bearded because females have to be big B to be bearded, and two will be yes, two will be unbearded, because the little b for females is dominant. Yes. Okay, then part D. Suppose a male and female, both which lack beards, produce a 1 beard to 1 no beard ratio among the male offspring. What are the genotypes of the parents? Okay, so dad is going to have to be, little b, to be unbearded. And mom is going to have to be big B, little b. What are the genotypes of the parents? Okay, so, and when we do that cross, it gives us, to undo this. Yeah. Okay. That's fine. And this one's lovely. Okay. So that will give us big B, little b up here. This will be little b, little b. This will be little b, little b. Okay. And that gives us, what should we look at? We're at males? Yeah. So it'll be, yeah, so these two will be bearded. These two will be unbearded, which gives us a 1-to-1 ratio, just like the question asked for. And that leads to question 5.
Cool.
Okay.
Thank you. So in question 5, we're going to be talking about X-linked inheritance. And so in cats, the orange coat color is determined by a little b allele, a black coat is determined by a big B allele, and the heterozygous condition produces a phenotype known as tortoiseshell. So the big B and little b alleles are X-linked. Suppose you cross an orange male cat to a tortoiseshell female cat. So if we do that, we will get, so we have an orange male, and then a tortoiseshell female. And so that will give us a tortoiseshell female, and then an orange female, and then a black male, and a orange male. If we cross those two. Yeah, so we have a 1 out of 4 chance for each of those. And so for B, this question concerns the trait describing in part A. So male tortoiseshell cats are very rare. So what is the genotype of a male tortoiseshell cat? So for this, we would be looking at a boy cat, that for some reason, while they were developing, they received 2 X chromosomes. And because of that, they're able to become tortoiseshell because they'll have a big B and a little b, which is what we need to be tortoiseshell. And that'll take us to part C. And so for part C, we have a male that, we're talking about fruit flies now. So there's a mutation that produces scalloped phenotype. And then, so we need to assume that the scalloped phenotype is a result of an X-linked recessive mutation. Indicate the genotypes and phenotypes of the prodigy produced when a reciprocal crosses are done. So for that, we're going to have the male who has it, and then a female who doesn't. And I mean, you can't actually see it. There we go. Perfect. And so with that, we're gonna have, perfect. So we're gonna have 1/2 of normal females and then 1/2 are normal males. So if we do the reciprocal one of it, we're gonna have a male that is not affected and a female that is affected. And since it's recessive, it needs to be homozygous recessive. And so when we do that, we're going to get, we're going to get 2/4 normal females and 2/4 scalloped males. And so with that, we can see that this is X-linked because when we do the reciprocal, they are, the males are opposite of what they were before. And so for part D, so for this question, it concerns the scalloped phenotype again. And we're going to assume that the scalloped phenotype is a result of an autosomal recessive mutation, indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done. So we have a recessive male and then a dominant female. And so for this, it'll give us 4 out of 4 or 100% big N, little n. And then if we do the reciprocal of that, having the male be dominant and then the female being recessive, we're going to get the exact same thing. We're going to get the 4 out of 4 big N, little n. So because of that, we can see that it just proves the fact that it's autosomal because we're getting the same thing for both of them. And that concludes our group presentation today.
Awesome.
Great.